Up to this point in this course, we focused almost entirely on small molecule drugs, compounds that are chemically synthesized, relatively small, and designed to fit into well-defined binding pockets on proteins. However, many disease-relevant targets do not have small molecule-friendly binding sites. These include cytokines, cell surface receptors, and protein-protein interactions. Biologics emerged to address these limitations. Rather than designing a small molecule to fit into a protein, biologics allow us to use proteins themselves as drugs, dramatically expanding what is considered druggable. In medicinal chemistry, the term biologic has a very special meaning. Biologics are therapeutic agents whose active pharmaceutical ingredient is a large, complex biomolecule, usually a protein produced by recombinant expression in living cells. Importantly, the therapeutic activity of biologics depends on their higher order structure, meaning their folding, disulfide bonds, and sometimes glycosylations, or the addition of sugar residues, or other post-translational modifications. On this slide, I want to give you a broad overview of what we mean by biologics and the range of therapeutic modalities that fall under that umbrella. At the highest level, biologics are therapeutic agents that originate from living systems. However, in medicinal chemistry, we use the term more narrowly than the phrase alone might suggest. What distinguishes biologics is that the active drug itself is a large, complex biomolecule, typically produced using recombinant biological systems, and its therapeutic function depends on the proper folding and higher order structure. The examples shown here highlight several major classes of biologics. Fusion proteins combine functional domains from different proteins to improve stability, targeting, or half-life. Peptides represent a smaller class of biologics that often mimic endogenous signaling molecules such as hormones. Antibodies, which we'll focus on in this lecture, are large, highly specific proteins designed to recognize extracellular targets with exceptional selectivity. I've also included CAR-T therapy here to show the full spectrum of what biologics can encompass, although we won't be covering cell-based therapies in detail. This is mainly to emphasize how far modern therapeutics extend beyond the traditional small molecule. Importantly, not all drugs that come from living organisms are biologics. Small molecule natural products, even though they may be produced from microbes or plants, are still classified as small molecules because their activity does not depend on protein structure or biological manufacturing processes. In the next slide, we'll directly compare biologics and small molecule drugs to make these distinctions clearer and to understand why antibodies behave so differently in terms of pharmacokinetics, targeting, and clinical use. This table highlights the fundamental differences between small molecule drugs and biologics at the big picture level. Small molecules are typically on the order of 500 Daltons or less. Because of their size and chemical properties, many small molecules are capable of crossing cell membranes and targeting intracellular enzymes or receptors. As a result, their design relies heavily on structure-activity relationships and chemical optimization strategies, such as QSAR. In contrast, biologics are much larger, often around 150 kilodaltons in size, in the case of monoclonal antibodies. They cannot cross cell membranes and therefore act almost exclusively in the extracellular space. This fundamentally changes the kinds of targets they can address, shifting the focus towards cell surface receptors, circulating ligands, and protein-protein interactions. Because biologics are proteins rather than small chemical entities, their design is driven primarily by target biology rather than traditional medicinal chemistry optimization. Instead of modifying functional groups, we focus on selecting the right biological target and engineering a molecule that interacts with it with high specificity. These differences explain why biologics and small molecules are not interchangeable. Each class is suited to different biological problems, and modern drug development often relies on both approaches in parallel. With this framework in mind, we'll now focus on monoclonal antibodies and examine their size, specificity, and extracellular mode of action that make them effective therapeutic agents. Antibodies are naturally evolved to recognize specific molecular targets with extremely high affinity and selectivity. Therapeutic antibodies exploit this natural specificity to block signaling pathways, neutralize ligands, or modulate immune responses. Because antibodies do not enter cells, they are particularly well suited for targeting cytokines, growth factors, and cell surface receptors that drive disease. This diagram shows the basic structure of an IgG antibody, which is the most common format used in therapeutic monoclonal antibodies. An antibody is composed of two identical heavy chains and two identical light chains held together by disulfide bonds. The heavy chains form the central backbone of the molecule, while the light chains pair with the heavy chain along the outer arms. At the tips of each arm are the variable regions labeled VH on the heavy chain and VL on the light chain. Together, these variable domains form the antigen binding site. This region is responsible for the remarkable specificity of antibodies and determines what molecular target the antibody recognizes. The upper portion of the antibody is referred to as the FAB region, or fragment antigen binding region. This is the part of the antibody that directly interacts with the target antigen, such as a cytokine or a cell surface receptor. The central hinge region provides flexibility, allowing the two FAB arms to move relative to each other. This flexibility helps antibodies bind targets that are spaced differently or arranged in complex environments. The lower portion of the antibody is the FC region, which is formed entirely by the constant domains of the heavy chains. The FC region does not determine target specificity, but it does play a critical role in the pharmacokinetics and immune interactions, including antibody half-life and engagement with immune receptors. From a medicinal chemistry perspective, you can think of the FAB region as controlling what the antibody binds, while the FC region influences how long the antibody persists in the body and how it interacts with the immune system. This structural organization helps explain why antibodies are highly specific, long-acting biologic drugs, and it sets the stage for understanding how therapeutic antibodies like Nemluvio function. One of the most common and clinically important mechanisms of antibody drugs is the blocking of the ligand-receptor interactions, thereby disrupting downstream signaling. On the left of this figure, you can see the normal biological situation. A ligand, often a cytokine or a growth factor, binding to its receptor on the surface of a cell, and that binding event initiates a signaling cascade inside the cell, ultimately producing some physiologic response. Antibody therapeutics interrupt this process in two primary ways. In the first case shown here, binding of a soluble ligand or a receptor. The antibody binds directly to the soluble ligand before it reaches the receptor. By occupying the ligand's binding surface, the antibody prevents it from engaging the receptor. As a result, the signaling event does not occur. Alternatively, the antibody can bind directly to the receptor itself, physically blocking the ligand from accessing its binding site. Even though the ligand is present, it can no longer activate the receptor, and downstream signaling is halted. On the right side of the figure, we can see a similar concept applied to cell-bound ligands or receptors. Here, on the right side of the figure, we see a similar concept applied to cell-bound ligands or receptors. Here, the antibody binds to a receptor or a ligand already associated with the cell surface. Again, this prevents the productive ligand-receptor interaction and blocks signal transduction. In both cases, the fundamental principle is the same. The antibody acts as a highly specific molecular shield, preventing the physical interaction required to initiate signaling. From a medicinal chemistry perspective, this is not enzymatic inhibition in the traditional small molecule sense. Instead, it's a steric blockade of a protein-protein interaction. The antibody's large surface area allows it to disrupt interactions that would be very difficult to inhibit with a small molecule. This mechanism is exactly what we'll see in the case of Nemluvio, which blocks IL-31 receptor signaling to reduce chronic itch. In the previous slide, we focused on antibody-mediated blockade of ligand-receptor interactions. This slide shows two additional mechanisms by which antibody drugs can function: mimicking natural ligands and recruiting immune-mediated cell destruction. On this side, you see the mimicking natural ligands. In some cases, an antibody doesn't block signaling. Instead, it mimics the ligand and activates the receptor. On the left side of this panel, you can see the normal scenario where a natural ligand binds to a receptor and initiates a signaling cascade. On the right, the antibody binds to the receptor in a way that induces a similar conformational change, triggering the signaling event in the absence of the natural ligand. In this context, the antibody functions as an agonist rather than an antagonist. From a medicinal chemistry perspective, this is analogous to small molecule agonists, but the mechanism differs structurally. Instead of fitting into a small binding pocket, the antibody stabilizes the receptor conformation through protein-protein interactions. While less common than blocking antibodies, agonistic antibodies are a powerful tool when activation of a pathway is therapeutically beneficial. On this side, we see the immune-mediated mechanisms of cell destruction. This third mechanism involves recruitment of the immune system through the antibody's Fc region. When an antibody binds to a target cell, its Fc region remains exposed. This allows immune effector cells, such as macrophage or natural killer cells, to recognize the antibody-coated target through the Fc receptors. This can trigger antibody-dependent cellular cytotoxicity, or ADCC, in which effector cells release cytotoxic granules that destroy the target cell. Antibodies can also activate the complement cascade, leading to complement-dependent cytotoxicity, or CDC, resulting in membrane disruption and cell lysis. Additionally, antibody-dependent phagocytosis can occur where macrophages engulf the antibody-tagged cell. These mechanisms rely not on blocking signaling directly, but on using the immune system as a secondary effector mechanism. Now let's apply everything we've discussed to a real therapeutic example, Nemluvio, whose generic name is Nemolizumab. On the left side of this slide, you see the structural model of a monoclonal antibody, the two heavy chains and the two light chains arranged in the classic IgG configuration. Nemluvio is built on this same structural framework. Like other therapeutic antibodies, its specificity is determined by the variable regions at the tips of the Fab arms. On the right side, you see the clinical product, a pre-filled injection pen. This highlights the important practical distinction between biologics and small molecules. Antibodies must be administered by injection because they are too large and structurally complex to survive the gastrointestinal tract or cross epithelial barriers efficiently. Nemluvio is approved for the treatment of moderate to severe atopic dermatitis and chronic pruritus. Its therapeutic effect is achieved through highly specific targeting of the interleukin-31 receptor, or IL-31 receptor. Interleukin-31 is a cytokine produced by activated T cells and is strongly associated with itch signaling. When IL-31 binds to its receptor on sensory neurons and skin cells, it initiates signaling cascades that contribute to chronic itch. Nemluvio binds to the IL-31 receptor and blocks IL-31 from activating it. Mechanistically, this falls into the first category we discussed, the receptor blockade. The antibody physically prevents ligand-receptor interaction and thereby disrupts downstream signaling. Importantly, Nemluvio does not broadly suppress the immune system. Instead, it selectively targets a signaling pathway specifically associated with itch. This illustrates how biologics can provide highly targeted modulation of disease pathways. From a medicinal chemistry perspective, Nemluvio represents a situation in which a protein-protein interaction, the cytokine binding to its receptor, is more effectively disrupted by a large surface-recognizing biologic than by a small molecule. In other words, the nature of the biologic target dictated the choice of drug modality. So before we move forward, I want you to watch a short mechanistic video from the pharmaceutical company. As you watch, I want you to focus less on the branding language and more on the biological pathway being illustrated. Specifically, pay attention to how IL-31 interacts with its receptors and how that signaling propagates through the different cell types. IL-31 is a cytokine, sometimes referred to as a neuroimmune mediator. It binds to IL-31 receptor alpha, which is expressed on multiple cell types. When IL-31 binds to its receptor, it activates sensory neurons in the skin and in the dorsal root ganglion. This amplifies itch signaling to the brain, which drives the urge to scratch. But IL-31 signaling does more than stimulate itch. It also interacts with immune cells, contributing to both T helper 2 and non-T helper 2 inflammatory pathways. It affects keratinocytes, which are the skin cells, interfering with proper differentiation and contributing to skin barrier dysfunction. And it stimulates fibroblasts, promoting collagen deposition and tissue remodeling, which can lead to fibrosis. So IL-31 is not just an itch mediator, it's participating in multiple interconnected disease processes. Now let's take a look at that video.
IL-31, the neuroimmune cytokine, directly drives disease processes by binding to IL-31 receptor alpha to activate sensory nerves in the skin and on the dorsal root ganglion to amplify the itch signal to the brain, which compels the patient to scratch, interact with multiple cell types to drive Th2 and non-Th2 inflammation, interfere with keratinocyte differentiation that leads to skin barrier dysfunction, stimulate fibroblasts to induce collagen deposition and tissue remodeling that result in fibrosis. Nemluvio is the first and only neuroimmune treatment that targets IL-31 signaling to work across 4 disease processes. Blocking signals in the sensory nerves and uniquely on the dorsal root ganglion to directly suppress itch. Blocking IL-31 receptor alpha to disrupt a key cause of harmful inflammation in the immune system. Blocking the IL-31RA on keratinocytes to normalize the skin barrier. And finally, Nemluvio blocks the IL-31RA on fibroblasts to inhibit profibrotic and inflammatory responses to help heal the skin.
Now that you've seen the video, let's reconstruct the mechanism ourselves. The central biological event is IL-31 binding to IL-31 receptor alpha on multiple different cell types. That single ligand receptor interaction drives 4 interrelated pathological processes. First, in sensory neurons, the dorsal root ganglion, IL-31 signals amplifies itch transmission to the brain. That neural activation compels scratching behavior. Second, IL-31 signaling promotes inflammatory responses through immune cell activation, including T helper 2 and non-T helper 2 pathways. Third, IL-31 disrupts keratinocyte differentiation, contributing to skin barrier dysfunction. And fourth, IL-31 stimulates fibroblasts, promoting collagen deposition and tissue remodeling, which can lead to fibrosis. What Nemluvio does is remarkably simple mechanistically. It binds to IL-31 receptor alpha and prevents IL-31 from activating it. That is receptor blockade. Nothing more complicated than that. But because IL-31 receptor alpha is expressed in multiple cell types, blocking a single receptor interrupts signaling across neurons, immune cells, keratinocytes, and fibroblasts simultaneously. This is an example of targeting a signaling hub rather than treating downstream consequences individually. Now let's step back and ask an important medicinal chemistry question. Why was an antibody the appropriate modality for this target? Interleukin-31 or IL-31 is a protein cytokine produced primarily by the T helper 2 lymphocytes. Structurally, it's a folded protein that interacts with its receptor through a relatively broad protein-protein interface. On the right side of this slide, you can see the structural representations of IL-31 and its receptor domains. Notice that the interaction surfaces are extended and involve multiple residues. There's no deep enzymatic binding pocket or small binding cleft that would be easily targeted by a traditional small molecule. From a drug design standpoint, this is a classic protein-protein interaction interface. These surfaces are relatively large and flat compared to catalytic enzyme pockets. Small molecules typically bind in well-defined cavities, but here, the biology presents an extended surface interaction between two folded proteins. An antibody, however, has a large complementary binding surface in its variable region. That makes it particularly well-suited to disrupt protein-protein interactions like IL-31 binding to its receptor complex. So the choice of an antibody was not arbitrary, it was dictated by the structural biology of the target. Now let's examine the pharmacokinetics of Nemluvio and connect it to our general antibody behavior. After subcutaneous injection, peak concentrations occur at approximately 6 days. This delayed Tmax reflects the slow lymphatic absorption, which is typical for large biologic molecules. The dosing regime includes 2 initial injections followed by once-monthly administration. The initial loading doses help achieve the therapeutic concentrations more rapidly, while the long half-life allows maintenance with once-monthly dosing. Compare this to a small molecule antihistamine with a half-life of 6 to 12 hours, requiring daily or multi-daily dosing. Drug modality directly affects dosing frequency. The volume of distribution is about 7.7 liters. That's relatively small and suggests that Nemluvio remains largely confined to the vascular and interstitial compartments. Unlike small molecules, it doesn't distribute widely into tissues or cross cell membranes. This diagram illustrates the key reasons why monoclonal antibodies like Nemluvio have such long half-lives. Antibodies circulating in the bloodstream are continuously taken up into cells through fluid phase endocytosis. Once inside endosomes, the environment becomes acidic. At this lower pH, IgG antibodies bind to a receptor called the neonatal Fc receptor, or FcRn, shown here in green. FcRn acts as a rescue receptor. Instead of allowing the antibody to be degraded in the lysosomes, FcRn binds to IgG and directs it into recycling endosomes. The antibody is then transported back to the surface of the cell and released into the circulation, where at physiological pH it dissociates from the FcRn. Any antibody that does not bind to the FcRn is sent to the lysosomes and degraded. It's degraded into peptides and amino acids. This recycling process dramatically extends the half-life of IgG antibodies. For Nemluvio, the elimination half-life is approximately 19 days, which is why monthly dosing is sufficient to maintain therapeutic concentrations. Notice how different this is from small molecule drugs. There's no cytochrome P450 metabolism, no renal filtration as the primary elimination route, and no active excretion into bile. Instead, elimination is governed by protein catabolism and FcRn-mediated recycling. This is a fundamental pharmacokinetic difference between biologics and small molecules. Antibody half-life is determined by biology, specifically Fc receptor recycling or lysosomal degradation, not by chemical stability or hepatic enzyme activity. So up to this point, we've focused on antibody-mediated receptor blockade using Nemluvio as an example. But antibodies are not limited to cytokine signaling pathways. The IgG structure we've studied, with its variable region for antigen recognition and constant region for immune engagement, enables a wide range of therapeutic mechanisms. For example, antibodies can bind and neutralize pathogens such as bacteria or virus. They can also block toxins or activate complement pathways. And antibodies can recruit immune effector cells through the Fc receptor interactions, promote opsonization, and enhance microbial clearance. In the inflammatory disease example, Nemluvio leveraged the FAB region to block a single interaction. In infectious disease, antibody therapy can leverage both the FAB-mediated recognition and Fc-mediated immune recruitment. This transforms antibodies from simple receptor blockers into programmable immune modulators. In our antibiotics section of the class, you learned that multi-drug resistant bacteria have evolved numerous mechanisms to evade or resist small molecule antibiotics, and that we have diminished discovery of new antibiotics to fight these newly acquired bacterially resistant strains. However, antibodies operate through fundamentally different mechanisms targeting virulence factors, promoting immune clearance, or delivering targeted payloads. This represents a shift from killing bacteria chemically to disarming pathogens and directing the immune system biologically. It offers an alternate strategy that can be deployed alone or in combination therapy with traditional antibiotics to help address the appearance of multi-drug resistant bacterial strains. So this is an exciting area of research. So far, we've focused primarily on the conventional IgG antibodies with 2 heavy chains and 2 light chains with distinct FAB and Fc regions. But the antibody platform is much broader than this single architecture. Researchers are exploring alternative immunoglobulin formats that naturally occur in different species and can be engineered for therapeutic use. On the left, we see the conventional IgG format. This classic Y-shaped structure with the FAB regions responsible for antigen binding and the Fc region responsible for immune engagement and recycling. Avian IgY is structurally similar, but it has differences in the constant regions and effector interactions that offer a novel immunoglobulin that can be produced much more efficiently at large scale than standard human monoclonal antibodies. Camelids, such as llamas and alpacas, produce heavy chain only antibodies. These lack a light chain, and contain a single variable domain capable of antigen recognition. Sharks possess a related format known as the variable new antigen receptor, or VNAR. And from these heavy chain only formats, we can derive what are commonly referred to as nanobodies, which are single domain antibody fragments that are much smaller than conventional IgG molecules. From a medicinal chemistry perspective, these alternative formats offer unique advantages. Smaller antibody fragments may penetrate tissues more effectively. They may access repressed epitopes that conventional IgG cannot reach. They can be engineered into multivalent or bispecific constructs, and because they are modular, they can be fused to toxins, antibiotics, or other payloads. So now that we've seen an example of a monoclonal antibody therapy with Nemluvio, and introduce some novel ways that antibodies are being developed as anti-infective agents, let's step back and compare monoclonal and polyclonal antibodies more broadly. Monoclonal antibodies are derived from a single B-cell clone. That means that every antibody molecule is identical and binds to the same epitope on the antigen. Because of this, monoclonals offer high specificity and reproducibility. Each manufactured batch behaves essentially the same way, but that precision comes at a cost. They are expensive to develop and produce. In contrast, polyclonal antibodies are a mixture of antibodies derived from multiple B-cell clones. Instead of recognizing a single epitope, they bind to multiple epitopes on the antigen. This provides broader binding coverage. If one epitope mutates, others may still be recognized. However, because they are mixtures, polyclonal preparations can vary from batch to batch, and when derived from non-human sources, they may carry a greater risk of hypersensitivity reaction. So in short, monoclonals give us precision, polyclonals give us breadth. In which scenario might breadth actually be advantageous? When might we want antibodies that recognize multiple epitopes instead of just one? Let's apply our question. When might breadth be advantageous? Rabies is one of the most lethal viral infections known. Once neurological symptoms begin, such as hydrophobia or fear of water, confusion, hallucinations, aggression, and paralysis, the mortality rate approaches 100%. The rabies virus is an enveloped RNA virus. Its surface glycoprotein is responsible for binding host cells and initiating infection. After a bite exposure, the virus replicates locally in muscle tissue. Then it travels retrograde along peripheral nerves to the central nervous system. There's a narrow window of opportunity before it reaches the brain, and during that window, we need immediate viral neutralization. This is not the time for slow adaptive immune development. This is the time for passive immunity. In post-exposure prophylaxis, patients receive rabies immune globulin. This is a polyclonal antibody preparation, meaning it contains antibodies targeting multiple epitopes on the rabies glycoprotein. That breadth increases the likelihood of neutralizing viral particles before they enter the neurons. In this case, breadth becomes life-saving. The product shown here is KEDRAB, a human rabies immune globulin preparation. This is a polyclonal IgG preparation derived from vaccinated human donors. It contains a mixture of antibodies targeting multiple epitopes on the rabies virus glycoprotein. After a suspected rabies exposure, this immunoglobulin is infiltrated directly into and around the wound site. Why? Because the virus is still localized in the muscle tissue before it enters the peripheral nerves. Once it enters neurons, antibodies can no longer reach it. In this scenario, breadth is advantageous. we do not know exactly which viral particles are present, and we do not have time for immune maturation, and we can't risk escape mutations. A polyclonal mixture increases the probability of complete viral neutralization. Rabies immune globulin is given together with the rabies vaccine. The immune globulin provides immediate passive protection, and the vaccine induces active immunity over time. Overall, antibodies are incredibly powerful therapeutic tools, but they are not perfect. Their large size limits delivery. They must be injected, and they often struggle to penetrate certain tissues, especially the brain. Furthermore, they must be cold stored and transferred to prevent degradation. Because they are proteins, they can also be immunogenic. Patients may develop anti-drug antibodies that either reduce efficacy or cause hypersensitivity reactions. Manufacturing is complex and expensive. Unlike small molecules, these are biologically produced and must be carefully purified and stored. And biologically, pathogens can mutate. Cytokine networks can be redundant. A single target monoclonal may not always be sufficient. While antibodies are elegant and precise, they're not simple solutions. They require strategic design and clinical context. So, if antibodies are expensive and complex, how do we lower the costs and increase access for more people? Unlike small molecule drugs, biologics cannot be copied exactly. These are large, structurally complex proteins produced in living systems. So instead of generics, we use the term biosimilars. A biosimilar must demonstrate high similarity to the reference product, with no clinically meaningful differences in safety, purity, or potency, but because of biological production variability, it's never molecularly identical. So for small molecules, generics are identical chemical structures. For biosimilars, even small changes in glycosylation or folding can alter immunogenicity or pharmacokinetics. So biosimilar approval focuses heavily on structural analytics, pharmacokinetics, immunogenicity, and chemical equivalence. So we've just spent this lecture talking about the power of biologics, especially monoclonal antibodies, but with power comes cost. Biologics are among the most expensive therapeutics in modern medicine, often costing tens of thousands of dollars per year. Biosimilars are designed to reduce that burden. A biologic may take 15 years and over $1 billion to develop. A biosimilar, in contrast, can be developed in 8 to 10 years at a fraction of the cost. They're not identical copies because biologics can't be copied the way small molecules can, but they're highly similar with no clinically meaningful differences in safety or efficacy. This matters tremendously in oncology and autoimmune diseases where monoclonal antibodies are foundational therapies. Overall, antibody therapeutics represents the evolution from chemical pharmacology to biological engineering. Biosimilars represent the next evolution, from innovation to accessibility.